All right, so we've been talking about memory. We've talked about um, many studies, some of the more difficult ones. This one on echoic memory, uh, this study on dichotic listening, uh, this stuff on the Sternberg task, especially the P300 latency aspect of the Sternberg study. Those are some of the more difficult studies we've talked about so far, and all of those could potentially be on your test number 2 coming up in a little over 2 weeks. Last time when we left off, we were talking about patient HM, and patient HM, can someone mute that microphone, please? Patient HM had showed this dissociation between declarative memory and non-declarative memory, as evidenced by the fact that he would not be able to remember things like if he met you today and then you saw him again tomorrow, he's not going to be able to remember that he met you the day before. He's not going to remember your name. Those are factual memories. And as a result of his removal of his medial temporal lobe, including the hippocampus in both hemispheres, he's unable to learn, to acquire, to encode new factual, that is declarative memories. He had anterograde amnesia, very, very slight retrograde amnesia. His primary impairment was for sure anterograde amnesia, the inability to encode new long-term declarative memories, though his procedural memory was intact. We know his procedural memory was intact because of that mirror drawing task, in which case every time that he would perform the mirror drawing task, he was getting better at it, as indicated by the quicker amount of time it took him. He would look in the mirror, he'd have to draw the reflection of what he's seeing, and he got quicker at that task over time. But each day he would do this task, you'd ask him, have you ever done this task before? He would not be able to remember doing the task before. He'd say, no, I've never done this before. So his mind, consciously, he was not able to remember the mirror drawing task, but his body was able to remember the mirror drawing task. So his non-declarative, his procedural memory, the ability to perform the mirror drawing task, was still intact, but his conscious, that is declarative memory, was not intact. It was impaired. So today we're going to start by kind of focusing more on the region of the brain that was removed in HM. Let's see, the hippocampus is this blue thing right here. It's in the median. This is whole thing right here is the temporal lobe. The hippocampus is this blue part right here. And HM also had some of the adjacent regions of the brain removed as well. So here we have the amygdala, pararhinal cortex right here, entorhinal cortex right here, parahippocampal cortex right here. We're actually going to talk about a lot of these brain regions in more detail today. We're not going to talk about the amygdala that much in this chapter. We will in the next chapter when we talk about emotion. But in this chapter, we're definitely interested in the hippocampus, pararhinal cortex, entorhinal cortex, and parahippocampal area. These 4 brain regions, all part of the medial temporal lobe right here, all were to some extent removed in patient HM and are partially responsible for his difficulty with forming new long-term declarative memories. So here is, I think, a quite helpful diagram that the textbook provides. It's kind of a hypothesis about how information is encoded into the hippocampus to allow us to create new long-term declarative memories. One of the things which is kind of cool about this diagram, hypothesis theory, which is presented here, is it kind of is in line with some of the stuff we talked about earlier in the semester. We've talked about a what pathway in the brain, also known as the ventral pathway. And we've also talked about a where pathway, which is also known as the dorsal pathway. The idea, which we talked about those chapters way back in 5, and those ideas we talked about way back in chapters 5 and 6. The idea is that in the brain, these types of information are at least partially separate. Some parts of the brain give you information about what it is you're interacting with, the identity of an object, the item that you are, or items, the objects that you are dealing with, versus where, which is telling you where are these objects out in the world, what do I have to do to interact with these objects. And so we see that there's kind of a similar type of separation of information as information is heading into the hippocampus. So that in this diagram, this part up here at the very top is the hippocampus. You do not need to know these subregions, CA3, CA1, dentate gyrus, subiculum. These are all subregions of the hippocampus that you will not be responsible for. Just understand that when you see these regions, and when you see like them all together like this means that they're all forming the hippocampus together. What we're really interested in this diagram is how information is being injected, is being sent in, it's being inputted into the hippocampus. And so there's really three brain regions which are involved in this diagram with inputting information into the hippocampus. We'll start with kind of the what side of things. This PRC, this stands for pararhinal cortex. And this right here, the PHC stands for parahippocampal cortex. Pararhinal cortex, PRC, versus parahippocampal cortex, PHC. Each of these kind of inputs into the EC right here, the entorhinal cortex. So I'm actually going to go back to our little brain thing. So the entorhinal cortex is right here. This inputs directly up into the hippocampus. So the EC right here inputs up directly into the hippocampus right here. Right here is the pararhinal cortex. The pararhinal cortex sends item information, what it is that you're looking at. It sends that information to the EC, the entorhinal cortex right here. There's also the parahippocampal cortex, which is sending where information to the EC. And then the EC, the entorhinal cortex, sends that information up into the hippocampus. So once again, some of the information from the pararhinal cortex is going to the EC, which is then sent up to the hippocampus. The where information is coming from the parahippocampal cortex to the EC, and then it's sent up. So there's kind of these two pathways which are converging on the entorhinal cortex. They meet there at the entorhinal cortex, and then the information is inputted up into the hippocampus. Two pathways kind of converge at the EC, and then they send their information up into the hippocampus. And then I'll point out, though, that the information that is hypothesized to be inputted into the entorhinal cortex from the pararhinal cortex is what information. So this is like, what is it that I'm trying to remember? What are the objects that I am trying to remember? The where information is inputted from the parahippocampal cortex. And I'm going to add some things. It's where information is one of the things which is inputted by the PHC. But I would say more generally, the type of information that is inputted by the parahippocampal area, parahippocampal cortex, could be contextual information, information about the scene in which I'm remembering something. So for instance, when I first went ice skating, I give you an example of my long-term declarative memory, my episodic memory from the very first time I went ice skating. And that memory involves objects that I was remembering. So for instance, I told you about my blue skate guards. I remember my blue skate guards. That's a specific object that I remember from that memory. My blue skate guards, which my parents left on my skate so that when I stepped on the ice, I immediately fell. And then, so that would be an example of an object, one of the items, one of the what's that I have stored in my long-term memory that was encoded to my long-term memory. But there's also information about where, and by where I mean the general context. And I've told you some of the contextual information was when I entered the ice that day, I entered from the corner entrance. Sometimes you enter from the side entrance if you were playing hockey, but that very first time I went ice skating, I entered from the corner entrance. I remember there's 2 rinks at that ice stadium, and that was on rink 1. So that's kind of the contextual information. There's some of the what's are some of the various items that were involved with that episode, like my blue skate guards, my ice skates. But then there's also some of the contextual information, like where was I when all this stuff was happening? And I was in Carmel, Indiana, rink 1. We entered through the corner entrance. That's the contextual information. That's kind of where the things happened versus what were the things that were involved. The what are the objects, the where involves kind of this contextual scene of where the memory took place. These 2 converging pathways both kind of meet at the entorhinal cortex and then both send their information up to the hippocampus. So here is some additional information. This was taken from a somewhat recent article in Current Biology, which is a pretty good journal, a journal article from Current Biology about the hippocampus. Oops. Um, and so, yeah, some facts here. These are kind of repeating some of the things that were mentioned here, but you can see that were mentioned on the previous slide. But you can see here the entorhinal cortex, the EC, that's sending information up into this thing up here. This thing up here is the hippocampus right here. This is an actual anatomical picture of an actual hippocampus in some type of animal. The EC, the entorhinal cortex, shoots information up into the hippocampus. Not shown here in this picture are neither the pararhinal cortex is shown nor is the parahippocampal area shown. But those two pathways would be sending information to the EC, which then sends information up into the hippocampus. And so the entorhinal cortex kind of represents both the spatial information, that is where, information about where, and the object information about what. The EC represents both of these types of information before it's sent up into the hippocampus. The pararhinal cortex, though, which I am definitely repeating from the previous slide, it inputs object information about what to the EC, whereas the parahippocampal cortex inputs, I'm just calling it more generally here, contextual information. On the previous slide, I called it where information. Where versus contextual information, I'm kind of using these terms interchangeably. And I'll give you some evidence supporting this claim regarding the parahippocampal area and this word study, which is from the textbook that I'll be talking about in a second. One thing I'll point out about the hippocampus and the entorhinal cortex, which are shown here, is they form what is called in this article the trisynaptic loop, which basically the loop part of this you can see here by the fact that the entorhinal cortex is inputting information into the hippocampus, but then the hippocampus is outputting information back to the EC. So there's a loop here. There's an input. The hippocampus is doing something to that information, and then it's outputting that information back to the EC. And there's kind of this loop of information. Information's sent into the hippocampus where it's somehow transformed, somehow processed, and then it's sent out of the hippocampus back to the EC. And we have this kind of loop of information right here. And together, these regions are doing something to help us form long-term declarative memories. So we're going to talk a little bit about place and grid cells. These are specific types of neurons which are found in aspects of, in particular, the entorhinal cortex, but also throughout the hippocampus. So the hippocampus is, these place cells are found in the hippocampus, and then these cells called grid cells are found in the entorhinal cortex. The reason why we care about these cells is they appear to be doing something about helping us encode into long-term declarative memory information about where. They're basically neurons that are found, well, we'll start with the place cells. Place cells are neurons that are found particularly in the hippocampus, and they appear to be important for representing where I am relative to the scene in which I'm remembering something. So this is giving me information about my location relative to the other things in the scene, relative to the situation that I find myself. So what is showing here, all of this right here represents a single neuron. This whole thing represents a single neuron. The square itself represents like a room. These little squiggly lines represents where an animal, it's probably a mouse or a rat, these little squiggly lines represent where an animal is moving in a room. And these little red dots, they show where this one neuron in the hippocampus, it becomes activated when the rat or the animal is moving at this exact location. So when the animal is moving over here, this particular neuron in the hippocampus is not being activated. But then as the rat approaches this part of the room, this one neuron becomes activated in the hippocampus. That's why it's called a place cell. It's basically a neuron or a set of neurons in the hippocampus that become activated depending on your location in a given scene, your location in a context. So right now I'm sitting in the corner of the master bedroom. Right now there's presumably one set of neurons in my hippocampus which are being activated right now. And then if I moved over here to my right is my couch. If I were to sit over there, presumably there'd be a completely different set of neurons that would then become activated. The idea is there's basically a set of, in a given scene, the scene here is the master bedroom. There's a given set of neurons which become activated for each possible location that I could be standing or sitting, each possible location in the master bedroom. This particular neuron, which was represented in figure A, when the rat was standing kind of in the middle, upper part of the middle of this room, a single neuron would become activated, but when the rat was standing over here, the neuron would not become activated. So this neuron is helping you encode something about my relative location, my location in a scene relative to the other things in that scene. And presumably this information is helpful for remembering where you were when something was happening, which is an important part of episodic memory. Grid cells are a little bit more complicated and they're a little bit more mysterious as to what they're actually doing. They are located in the entorhinal cortex. Remember, the entorhinal cortex is kind of that place in the middle, and the entorhinal cortex sends up information up to the hippocampus. In fact, I want to show you the entorhinal cortex again. The entorhinal cortex is this one right here. It takes information from the parietal cortex and from the parahippocampal area, and it combines those information and then sends it up to the hippocampus, and the information from the hippocampus comes back to the entorhinal cortex. So the entorhinal cortex is part of like a recurrent loop. Recurrent meanings that it both inputs information, but it also receives inputs. It sends out information, but it also receives back information. That's what I mean when I said the entorhinal cortex involves recurrent processing. It both sends inputs, but it also receives inputs. But the entorhinal cortex has these grid cells. So a grid cell is shown right here. And grid cells are a little bit more complicated than place cells because a place cell, which is shown right here, it only responds. A given neuron, if it's a place cell, it only responds when I'm in one specific region of a context, one specific region of a scene. Grid cells, on the other hand, they respond, a given grid cell responds when I'm standing in many regions in a scene. So right now I'm in the corner of the room. And let's say that this is a grid cell inside my brain right here, and I'm in this corner of the room right here. If I were to take a few steps that way, then the grid cell would start to fire. If I walk even past that, so if I start walking towards the bed, which is over there, then the grid cell will turn off. But if I continue walking even further, when I go all the way to the nightstand, which is over there, then the grid cell turns on again. So a single grid cell represents the entire room like a grid. The grid cell does not respond when I'm right here, but if I move over here, the grid cell does turn on. If I move over here, it turns off. If I move over here, it turns on again. If I move over here, it turns off. And you can see why it's kind of called a grid cell. It kind of represents the different spatial locations like a matrix. It's not really clear why it's doing this. Place cells, yeah, it seems pretty clear. Place cell is representing my location, and a given place cell is representing a specific location in a scene, in a room. Grid cells, though, are more complicated because a single grid cell, this all represents one single grid cell. But a single grid cell actually represents many different regions of the room, but it does so kind of in this on-off fashion. It doesn't respond here, but it does respond when I'm over here. It doesn't respond here, but it does respond when I'm way over there. And you can kind of see the grid-like properties of grid cells right here. So these are mysterious, but somehow both of these types of cells appear to be working together to help encode contextual information. Here are the objects which are involved with in a long-term memory. Place and grid cells tell you information about where are the objects located, where are they located relative to me. So it's giving you kind of a representation of space. Questions or comments about place and grid cells?
Yes. So is each of the so in B in the grid cell thing, is each of the red dots a cell? Is like the whole square is the cell thing?
Well, yeah. So it's like each of the red parts represent the parts in space where a single cell becomes activated. So like imagine a single neuron in your brain. When I'm standing here, that neuron does not fire action potentials. When I'm standing here, the neuron is firing action potentials. When I'm standing here, the neuron is not firing action potentials. And when I'm standing here, it's firing action potentials again. So the whole thing represents, can you please mute that microphone, by the way? So the whole thing represents a single neuron, and the whole thing represents a single room. The red dots represents those parts of the room where that single neuron fires action potentials, and the parts that are not red represents the parts of the room where that single neuron does not fire action potentials. Any other questions or comments? Hopefully that answers your question there. Any other questions or comments about place and grid cells? Cool. Let's move on. Yeah, we're talking about some of the more complicated stuff in the brain. Placing grid cells, quite mysterious, quite important though. So here is one of the classic paradigms in animals for studying declarative memory. So the caveat or an important thing to remember though is, declarative memory we think about in humans in terms of the ability to verbalize the memory. Obviously animals don't talk. They don't verbalize. They don't state facts. But animals definitely have hippocampi. They definitely have a hippocampus in the left hemisphere. They have two hippocampi. And the question is if animals can't talk, so they're not verbalizing, but they still have memory, and they still have hippocampus. So what exactly is the hippocampus doing for animals? And to remind you that we already know that in humans, and in animals, we found these place and grid cells. We know that these place and grid cells are doing something to help us remember the spatial information about a long-term declarative memory, kind of those pictures in your head of where I was located and where the various objects were located in a given scene when I was forming a long-term declarative memory of that scene. And so researchers have been using this thing called the Morris water maze to kind of study the spatial aspects of animals' memory. Once again, it's not just to be measuring animals' declarative memory, but you just got to remember animals don't actually verbalize their memories. But researchers still assume that this is declarative memory that we're studying here in animals. So the way that the Morris water maze works is that you put the rat in this water maze, this little thing right here, and you can see right here is a platform. The animal, the rat, is highly motivated to find that platform because the water maze, the pool basically that it's in, is the water is taller than the animal's head. And obviously the animal doesn't want to have to swim all the time because it uses energy. The rats don't really like water to begin with, and they definitely don't like water when it's above their head because of course they're at a high risk of drowning. So the rats are highly motivated to find the platform because they can stand there. They don't have to swim. Their head can easily be above water. And so the researchers they use kind of this setup right here. And typically the dependent variable is how long does it take for the animal to find the platform? The idea is that you give the animal a lot of exposure to the Morris water maze. Eventually they start to form a long-term memory of where is the platform located in this room. An important thing to notice is that they're kind of these, we call these markers or something, there are these little points on the wall, like this green triangle, the red circle, the blue square right here. And these are kind of giving the rat contextual information. They're giving the rat information about where the platform is relative to the room, the context that the rat finds itself. For instance, right here, this particular picture right here, the platform, if the rat is being smart, if the rat is forming a long-term declarative memory, the rat's going to have some type of a memory being like, okay, the platform, this thing that I want to find, it's kind of between the red and the blue objects. It's between the red circle and the blue square. So if I want to find the platform, I want to kind of find the red circle and the blue square, and I want to swim between those things. And if I do that, then I increase my likelihood of finding the platform, which is a good thing. So the rat uses these markers on the wall to kind of find its bearings where the platform is. So the object, the item that the animal is trying to learn about is the platform. The where information is on the wall. That kind of gives the animal a bearing, like a sense of where it is relative to the platform, which is the object that it's trying to find. Let's see. Right. So the way they've used the Morris water maze is usually they will train the rat to perform. They'll drop the rat into the water maze many times, and they'll do so with either control rats whose brains are intact, or they'll have experimental rats whose different parts of the brain are removed. Different parts of the brain are lesioned or destroyed or just removed. And they want to see, how does removing these various parts of the brain influence the amount of time it takes the rat to swim to the platform. So there's a wrinkle here. There's a nuance about the findings. We know the hippocampus is important, but the hippocampus is only important for kind of one specific situation. I'll jump to that here. If the rats are if the rat is dropped from a different spot every single time it's placed into the Morris water maze, if their hippocampus is destroyed, then this is when things are really difficult for the rat. The rat whose hippocampus is lesioned, is destroyed, if they're placed every day in a different location, they really struggle to find the platform. Presumably, the reason is because if your hippocampus is destroyed, you're not able to form new long-term declarative memories. And those long-term declarative memories appear to be something along the lines of, the item that I'm trying to remember is the platform, and it's located contextually, where in space, it's close to the blue square. Animals whose hippocampi are lesioned, they need that contextual information on the wall. They need to remember the blue square is next to the platform. And so if your hippocampus is removed, though, you're not able to encode that information into long-term memory, which means you're going to have trouble finding the platform. The kind of wrinkle, the thing which makes this a little bit complicated, though, is that if you drop the rat in the same exact location in the Morris water maze every single time, they actually are able to find the platform no problem. However, the reason they're probably able to find the platform has to do with the fact that even when their hippocampus is lesioned, their procedural memory would be intact. If you are dropping the rat in the same location of the Morris water maze every single time, the rat can form a procedural memory where it says, okay, swim to the right for 3 seconds, and then I'll be at the procedure, or then I'll be at the platform. But they're using a procedural memory, just swim in that direction for a few seconds, and then we're good. It's when you drop the rat in a different area where then they have to look up. They have to use those markers on the wall and be like, oh yeah, the platform is over there by the blue square. I'm dropped way over here this time. I need to look up to the blue square, and then I need to swim over in that direction because that's how I'm going to find the platform. So hippocampal lesions don't hurt the rats when they're dropped from the same location every time because they can use the procedural memory. If you're dropped from a different location every time, though, the rats have problems with this because they're not able to use the long-term declarative memory that they formed. Questions or comments about the Morris water maze and how it's been used to study hippocampus function in animals? All righty. Okay. So this was a study that I mentioned earlier. This is a study that gives more information about both the hippocampus and the parahippocampal area and how they contribute to the formation of long-term declarative memories. This one's in humans again, though. I really want to focus your attention on the fact that we're involving the parahippocampal area. Earlier, we talked about the idea that the parahippocampal cortex is involved with inputting where information. And I kind of mentioned that when I say where information, I'm kind of using that interchangeably with the more general term contextual information. And so this is a study where they focus on the contextual information aspect of things. And you should I feel like you should write this down now or contribute or, really focus on forming a memory of this. The type of contextual information that was used in the study was color. They have to, in this study, the people had to remember the words that they saw, but they also had to remember contextual information about those words. That contextual information was, okay, yes, I saw the word nickel at one time in the past, and when I saw it, it was green. Similarly, they would see words like deer, you'd have to remember that. And you would also have to remember, okay, when I saw deer earlier, it was written in the color red. So the people in this study, they were presented with lists of words on day one. It was like a two-part study. On day one, they came in, they were presented the list of words. Sometimes some of the words were written in green. Some of the words were written in red. That was day one. During day one, this is like encoding, they were measuring these people's brain responses. I believe it was an fMRI, functional magnetic resonance imaging. So they're measuring the regions of the brain that were being involved when people were remembering what these words were, and also that the color that these words were written in. So that was day one. Basically, in day one, you're studying these words. You're trying to remember what words you saw. And really importantly, you're trying to remember the contextual information about those words. The contextual information is, what color was the word when I saw it? On day two, you come back into the laboratory again, and the researchers ask you questions which probe your memory of what you saw on day one. And there's really two things that the researchers asked you. They asked you on a scale of 1 to 6, how familiar is a given word? So on day two, they show you a word like nickel. And a word like nickel, that's a word that you saw on day one. So you would say something like, that is definitely an old word. This is a 6. That word is very familiar to me. It is definitely old because I saw it on day one. So that's judging the familiarity. You're judging how familiar, or here they call it how old it is. Very familiar means that it's definitely old. It's definitely a word that you saw during day one of the experiment. So that's the first question they asked you. How familiar is a given word? But they also ask you what's known as a source judgment. This is the question which the researchers are trying to figure out. Was the participant able to remember the contextual information about the word that they remember? So you might remember, yeah, I definitely saw the word nickel on day one. But then when they ask you, what color was nickel? You might be like, is that green or was it red? Or was it blue or was it purple? That's the context. That's the source judgment. That's asking you information about the context in which the word was originally presented. And so that's the second question they ask you, basically, what color was the word when you saw it? So if I see the word nickel and I say, yeah, that was very familiar. I give it a 6 out of 6. I definitely saw it on day one. I definitely recognize that I saw that word before. The second question they would ask you then is, what color was it when you saw it? That's the question, which is making sure that you are able to perform the source judgment. That is, you're able to answer information about the context in which that word nickel was presented. If you're able, so recollected this is kind of like a term that they use in the textbook and in this particular study. Recollected means it's a combination of two things. If I correctly say that, yes, I saw this word before, it is familiar to me, 6 out of 6. And if I get the answer correct regarding what color the word was presented originally, if I get both of those pieces of information correct, then that means that is a successful recollection. And what they're interested in this study, remember, they were measuring brain responses on day one. Day one, you were studying the words. And what they're interested in is, what does the brain look like? When you're encoding words on day one, what does the brain look like on day one for words that you later successfully are able to recollect? What does the brain look like for words that you later are able to successfully recollect, compared to those words that you are not able to successfully recollect later on? Some of the words on day two you get right. Some of the words on day two you don't get right. On day one, you saw all of these words and we're measuring the brain response. How does the brain response on day one to those words that later on we're going to get right? We're going to recollect them. How does the brain's response on day one? What does it look like for those words that later on we are not going to remember, those words that we are not going to successfully recollect? In other words, how does the brain encode words that we are successfully recollect compared to the words that we successfully do not recollect? So here are the findings of the study. Actually, this is probably the most, this is verbally explaining what's shown on right here. Words that were later recollected, that is words during phase 2, day 2, that you said, yes, that was familiar, and you got the color correct. That's a correct recollection right there. The words that were correctly recollected on day 2, when you were measuring the brain's response to those words on day 1, those words showed more activation in the hippocampus and the parahippocampal area. And so what this suggests is that these two regions of the brain, we already knew that the hippocampus was important for long-term declarative memories, but this reinforces the idea that the parahippocampal area is particularly important for encoding information about context. Once again, I really want you to remember the contextual information in this study was the color that the words were presented in. Those words that were later correctly recollected, judged as familiar, and the color was correctly recalled, showed higher activation in the hippocampus and the parahippocampal area. So this one's it's another challenging study, and it is highly likely I will be asking about it on test 2. Any questions or comments about it? Okay, moving on. I believe this is the last topic in this chapter. And this is consolidation theory. So consolidation theory explains some really interesting findings regarding the hippocampus and its involvement with the retrieval of long-term memory. So we already know, we've seen several pieces of evidence that suggest that the hippocampus is important in encoding long-term memories. We already know that. We've seen several pieces of evidence that the hippocampus is important for encoding long-term memories. HM was some of the evidence, and then we just saw the evidence in that word study previously, the fMRI study. It's also known, though, that the hippocampus is involved with retrieval of freshly formed memories. So I form a memory today of something. Someone introduces me to them. I get introduced to a new person today, and then they tell me their name. And then tomorrow I see that person again. And then I meet that person and I want to remember their name because I don't want to seem rude. And if you are measuring my brain activity while I'm trying to remember the next day the person's name, you're going to see some hippocampal activation. The reason is because although the hippocampus is definitely involved with encoding new long-term memories, the hippocampus is also part of retrieving those long-term memories when they're newly formed. So a day later, after you just learned someone's name, you're trying to remember that person's name a day after you met them, the hippocampus is going to be important in your ability to retrieve that person's name. The really interesting finding, though, is that the involvement of the hippocampus actually declines in retrieval over time. So I meet a person today. On day 1, when I meet that person again and I try and remember their name, lots of hippocampal activation. But if I see that same person like 2 months later, I still remember their name. But if you actually look at the activation of the hippocampus, I see that person 2 months later, 60 days later, and I'm able to successfully retrieve their name from my long-term memory. But if you actually looked, if you measured with like an fMRI or whatever, of what regions of my brain were most strongly activated 60 days later after forming this long-term memory of the person's name, the hippocampus is not strongly activated. So this is kind of weird. The hippocampus is activated in retrieval of long-term memories shortly after that long-term memory was formed, like a day later. But if you look at like a month or 2 months later, the hippocampus is no longer involved with the retrieval of that memory. And it turns out, and it mentions here, usually it's other outer cortical regions of the brain which do become involved. The hippocampus, the medial temporal lobe, is important for retrieval of long-term memories when those memories are fresh, when they just were encoded. The more time that passes, I kind of think of it, and other researchers think of it as kind of a migration of the memories, where at first the memory is stored in your hippocampus so that when you're trying to remember it, you use the hippocampus for retrieval. But after time, the memory moves and it starts to be stored kind of in a distributed fashion across the outer cortical regions of the brain, kind of the top parts of your brain. So that once the memory moves here, the hippocampus is no longer important for retrieving the memory. This makes sense because we've talked about with HM, he had very slight retrograde amnesia. Remember, retrograde amnesia is the inability to remember things that just happened. The things that he had trouble, when his hippocampus was removed, the stuff that he had trouble remembering was, as far as things that happened in the past, was things that had just been freshly formed memories. If you recall last time I said, he was able to remember the names of his parents. He was able to remember the name of his elementary school. He was able to remember all of the stuff that happened to him when he was a kid. The reason is because those long-term memories, those were stored in the outer cortical regions of his brain. Those remained intact. But the freshly encoded memories in the hippocampus, those memories were gone. And they would never come back because the hippocampus was removed. So this is consolidation theory, and I think I have a good picture. I have an experimental evidence for consolidation theory in animals, and I have kind of a nice picture that summarizes this idea coming up. So fear conditioning. This is one of the ways that consolidation theory has been studied. Basically, the animal starts to form an association between a particular environment, like a particular room, like they can be in either cage A or they can be in cage B or cage C. They're different rooms for the animal. And the animal learns from previous experience that when I step in environment A, when I'm in cage A, I get a shock. And so the behavior, if you have an intact brain, the behavior, if you're the rat with the intact brain, when they drop you in the environment, when they drop you in the cage where you had been shocked previously, the typical behavior is called freezing. Basically, the animal is placed in the room that they're scared, and they just freeze because they're scared. And they don't want to explore this area. Usually rats want to explore the area. But when they associate that area with a bad outcome, when in the past something bad had happened to them, then they are very unlikely to want to explore. They try and protect themselves by freezing. And so you look at an animal's memory. If the animal is able to form the long-term memory, this place is bad. I got shocked here previously. What you look at is the animal's moving behavior. If the animal remembers this is a bad place, the amount of movement is going to be small. If the animal failed to remember, oh, this is a bad place, I want to be careful here, then you're going to see the animal moves around a lot in that area when they're placed in that room again. So we look at the animal's moving behavior as a function of whether their hippocampus is intact or whether the hippocampus has been destroyed. So they train animals during this task. They put the animal in these different cages. One of the cages will shock them every time. They do that while the hippocampus is intact. Then for half of the animals, they destroy their hippocampus with some type of an injection. It's kind of a foggy figure, I'm sorry, but the name of the chemical that they inject is CNQX. I don't actually know what that acronym stands for, but this is a drug that when you inject it into the animal, it just basically turns off their hippocampus. And the dependent variable here is freezing behavior. So freezing behavior, if this is really high, that means the animal is stationary. When this bar gets low, like it is here, that means that the animal is exploring around the room. And remember, if the animal is able to remember, what they should be doing is freezing. They shouldn't be moving around a lot at all. So another independent variable here, we see that there's a two-way interaction here. There's two factors. One factor is whether, is the type of drug they're given. Saline versus the one that destroys the hippocampus. Saline versus the one that destroys the hippocampus. So the saline condition is like the placebo or the control condition. Those rats, hippocampi are fine. The ones in the CNQX condition, those rats, their hippocampi, they are destroyed. And we want to know, how important is this hippocampus for the retrieval of these fear conditioning memories? And another independent variable in this study is, when does this test take place relative to the learning phase? So in the learning phase, they're learning about the association. Cage A causes me cage A shocks me every time. So I'm scared of cage A. That's what they learned. Then they measure their freezing behavior 1 day after the animal learned this about cage A, about environment A. And then they also test them again 28 days later. And they want to see, how important is the hippocampus 1 day after you learned about environment A shocking you versus 28 days after learning about cage A shocking you. And what they show is that, so these rats right here, 1 day later, their hippocampus was destroyed. So they were not able to use their hippocampus to retrieve information about fear conditioning, that environment A is shocked them in the past, therefore they should be scared, they should be freezing. Like the control rats with saline, they freeze. But these rats here with their hippocampus lesioned on day 1, they don't freeze very much because they're not able to use their hippocampus to retrieve the information about environment A, and therefore they don't realize that they should be scared, so they do a lot of exploring here. But the really interesting thing you see here is on day 28, there's actually no difference in the rat's performance, or not a significant difference at least, between the control rats. They had their hippocampus was intact. The CNQX animals, these rats' hippocampi were destroyed. But you can see that it doesn't matter for these rats. These rats show a lot of freezing behavior, the red bar, they still show freezing behavior on day 28, because by day 28, your hippocampus is no longer important for retrieving memories. There's some type of consolidation that is occurring. And the more time that passes since you've encoded a long-term memory, the less important the hippocampus gets for retrieving that long-term memory. After 28 days, all that memory about fear conditioning, about environment A, cage A causing you to be shocked, that moves to the rat's outer cortical regions. The hippocampus is no longer important for retrieving that information. And so the rats here, even though their hippocampus is destroyed, they still show freezing behavior because they are successfully able to retrieve the memory using their outer cortical regions. So this is, once again, we're talking about consolidation theory, and this is behavioral evidence in rats that support consolidation theory. When your hippocampus is destroyed on day 1, you cannot retrieve the memory from about fear conditioning. On day 28, even though your hippocampus is destroyed, you still are able to retrieve the information about fear conditioning. And so here is kind of a schematic or a theoretical diagram, which is attempting to explain what's going on in your brain for the retrieval of long-term memories when it's only been several hours since you formed those long-term memories, compared to when it's been several weeks since you formed those long-term memories. This thing over here you can kind of ignore. The two things that really care about are these kind of connection maps right here, the hours one and the weeks one. These are the two important things to pay attention to here. And you can see that they're different. The connections are different, and the qualitative difference for how the connections are different explains or is really important to understanding this consolidation theory. So I kind of think as the hippocampus is kind of like a conductor, and when you have a freshly encoded long-term memory, remember the hippocampus is necessary to retrieve that long-term memory. The reason the hippocampus is important to retrieve a freshly encoded long-term memory is because the hippocampus appears to be sending signals to the cortical regions of the brain, and you can see that here. This is when I'm trying to retrieve a memory several hours after it's been encoded. The hippocampus is activated, and it's also sending activations to this cortical region. We'll say this is the frontal lobe. this cortical region, another region of the frontal lobe. And this cortical region will say this is part of the parietal lobe. So the hippocampus is sending signals to all these different outer cortical regions up here. And when I'm trying to retrieve a long-term memory that I just formed, my hippocampus is kind of organizing the firing, the action potentials that are being fired by all these outer cortical regions up here. The hippocampus is kind of the start of the process, and then it sends signals to these other cortical regions of the brain. And then the cortical regions of the brain and the hippocampus are kind of working together, with the hippocampus conducting or kind of organizing the firing pattern of the outer cortical regions of the brain. They're doing all of this to help you retrieve a freshly encoded long-term memory. However, several weeks later, connections have formed between those cortical regions of the brain. So we have, one frontal lobe region of the brain, another frontal lobe region, and maybe a parietal lobe region of the brain. Over time, for the past several weeks, those connections among these cortical regions have started to strengthen. They started to find synaptic connections over time. One of the theories about how consolidation occurs in the brain is that these regions of the brain that don't have connections at first, as the long-term memory becomes consolidated, connections form amongst the cortical regions. And then eventually, you can see, by the way, there's the red lines that are occurring between the cortical regions here. There are no red lines here, but there are red lines here. These red lines represent the fact that after several weeks, these functional connections have been formed between these cortical regions of the brain. And so as a result, several weeks later, you do not need the hippocampus to send signals anymore, because those signals can now be shared via these connections that are formed among the cortical regions of the brain. Here, after several hours, you need the hippocampus to send signals to all the cortical regions of the brain. All these different cortical regions need to be activated, and it's the hippocampus which is conducting, which is organizing the pattern of activation, because there are no connections between these cortical regions. But because of consolidation, which takes time, eventually connections can form amongst those cortical regions right here, such that the hippocampus is no longer necessary as a conductor. The hippocampus does not need to organize anymore, because these connections among the cortical regions have formed by themselves. So that's the idea of consolidation theory. And that's it for chapter 9 stuff. So you guys can get out of here if you want. We will be starting chapter 10 next week. We might be able to get to some chapter 11 stuff before the course is done. But yeah, we only have a couple weeks left to go. It looks like 3 more lectures if I'm counting correctly. So you guys can get out of here. If people want to stick around and ask questions, we definitely have time. I'm happy to talk about Master and his emissary papers as well if people want.